Hello and welcome to section 4.6 about mitosis. This is a long section, so I'm going to try and get through it quickly so the video doesn't get too long. We're going to start by examining what are chromosomes. If you'll remember when we talked about cells, the basic structure and function of all living organisms, cells contain an organelle called a nucleus. Inside the nucleus we find chromosomes. Chromosomes, right now, you need to understand that chromosomes contain genetic information. If we were to unravel this chromosome, it would contain a DNA structure, and pieces of that DNA structure are what we call genes. And we'll talk more about that later. For now, you know the chromosome is located inside the nucleus. The parts of a chromosome are shown here. This right here is a duplicated chromosome. If we were to have only a single chromosome that is not duplicated, it would just be one of these chromatids. Right here, we have a duplicated chromosome, and we have one chromatid, two chromatids. They are called sister chromatids. They're held together in the middle by what we call a centromere. So how do eukaryotic cells reproduce? They reproduce by mitosis. Remember, eukaryotic organisms belong to the kingdoms Plantae, Animalia, Protista, and fungi. Prokaryotic cells belong to the bacterial kingdoms, and they produce by a different mechanism called binary fission. And we're not going to really go into that right now. We're going to talk about mitosis, which is the eukaryotic cell reproduction cycle. And cell division, mitosis, is the process that happens in between interphase for the cell. So we're going to just start by talking about interphase in the next slide. And if you'll remember, here is interphase. It's most of the cell cycle, and it's where the cell carries on its normal processes as well as duplicates its DNA. Mitosis is this little part right here, and we're going to go into detail about each of these steps. So the stages of mitosis technically are prophase, metaphase, anaphase, and telophase, or telophase, however you would like to say it. Interphase precedes mitosis. It's the phase after mitosis. Cytokinesis is the process immediately following cytosis. It is simply where the cell's cytoplasm splits. So you may see these all as referred to as stages of mitosis. Technically, only these four are, and an easy mnemonic to remember them. P-M-A-T, PMAT. That will help you remember prophase, metaphase, anaphase, and telophase. So let's start with interphase, which is technically not part of mitosis, but it is an important part of the cell cycle. Before we get to mitosis, it is where the cell carries on its normal activities in the G1 and G2 phases. In the S phase of interphase, remember, the cell replicates its DNA. Here we have unreplicated strands of DNA, or chromosomes. Here, they have been replicated. We have replicated DNA, and remember, these are called sister chromatids. This happens in interphase. When we begin the prophase segment of mitosis, these are the things that happen. The chromosomes actually become visible. They condense. So when we're in interphase, the chromosomes kind of look like this. They're not very distinguishable. But as we enter prophase, we start to actually be able to see individual chromosomes because they condense. In prophase, the nuclear membrane breaks down, and spindle fibers form and attach to the centromeres of the chromosomes. So right here are some spindle fibers. They're beginning to form between these centrioles, if the organism has centrioles. These are the three steps that happen, the three things that happen in prophase. In metaphase, let's come on down to this phase in mitosis. Chromosomes now are pulled into a line along what we call the metaphase plate. It's like an equator. As these two regions, these centrioles, continue to pull apart, they come to opposite poles. These spindle fibers attach to each one of the chromosomes, the duplicated chromosomes at their centromeres, and they are pulled into this line right along the equator. So the lining up of the chromosomes along the metaphase plate or the equator is what happens in metaphase. The next step is anaphase. And during anaphase, the sister chromatids are pulled apart. They separate, and the spindle fibers that they are attached to begin to shrink back toward the poles, and the two sister chromatids actually separate and begin to pull apart. During telophase or telophase, the nuclear envelope reforms around each of the groups of chromosomes, and two new nuclei are formed. Cytokinesis follows the PMAT stages of mitosis. It's immediately following, and it is simply the division of the cytoplasm, cytokinesis. When that happens, we get two identical new daughter cells. They have the same chromosomal makeup, the same DNA in each one, and that is what is formed from mitosis. So let's look at how plant and animal mitosis is different. One of the main differences is that in cytokinesis, the process happens differently because of the cell wall in plants. So when animal cells undergo cytokinesis, they form what is called a cleavage furrow, right here in that picture, and then the two cells basically just pinch apart, forming the two new daughter cells. In plants, a cell plate is formed in between, and that ends up dividing the two cells so that they become two new cells due to the cell plate. Now, before I ask this question, just something to note is that for the most part, most organisms, most eukaryotic organisms undergo these processes very, very similar to this. Some eukaryotic organisms, their mitosis process varies just a little bit from this, but this is basically the fundamental, these are the fundamental steps of mitosis, PMAT. I would like you to right now pause the video and see if you can identify the stage of mitosis that some of these cells are experiencing. Here are some animal cells, here are some plant cells, looking at them under a microscope. See if you can identify which stage each of those is experiencing, and then the answers will be on the next slide. So here are the answers. If you can tell right here, this is interphase because we cannot distinguish the different chromosomes in there. They have not yet condensed, so they are still in interphase. They're not distinguishable. Here we have prophase. The chromosomes have condensed and they are starting to be visible, and we can see them. Here we have metaphase, where the chromosomes are lined up along the equator or on along the metaphase plate. Here we have anaphase, where the sister chromatids have separated and they are beginning to pull apart to opposite poles. And then here we have telophase, where we have two new nuclei being formed and cytokinesis is about ready to begin. So right here, the same things are visible in a plant cell, or in various in a plant organism in its various cells. A is metaphase because you can see them lined up along the plate. Here is anaphase. The sister chromatids are separating to opposite poles. Here we have telophase, where we have two new nuclei that have been formed and the cell is not yet divided through cytokinesis. E is interphase because we can't distinguish the chromosomes yet. They haven't condensed. And D here, we have prophase because we've just been able to begin to distinguish the different chromosomes. Ideas for advanced proficiency. Any questions or interests you may have, please research some of that about mitosis and cell reproduction. Here are just some different ideas that you can read about and see if you would like to research some of those for an advanced proficiency or a score of 4. So I look forward to seeing you in class, and discussing this topic further.